In the next 3 videos, we're going to look at how proteins are purified and how we analyze proteins that are purified. So we're going to talk about what you need to keep in mind environmentally when you purify proteins. We're going to look at assays that we can measure protein purification with. We're going to look at different techniques that we can use to purify proteins. That's including salting out. Also its ionic charge and polarity through chromatography. We're going to look at gel electrophoresis and how we can purify proteins using ultracentrifugation. Now, historically, if you want to study a protein, you need to remove it from the cell. And I put generally here because there are techniques to study a protein inside of a cell, but it is extremely hard to do. So you generally want to remove a protein from a cell to study it. Now, there are some negatives to this because if you want to learn about a protein, proteins don't work alone, so you really do want to study it inside of a cell. But like I said, we are not to the point yet where we can study a protein inside of the cell easily. So you want to do it outside the cell. So at the very beginning of biochemistry, we didn't have protein purification techniques like we do today. So you wanted to study proteins that were high in concentration, that being hemoglobin. So we'll see later on in this course when we talk about various protein interactions, our model protein is going to be hemoglobin because that's what everybody studied back in the day. However, as we talked about in previous videos, we now can do molecular cloning. That is, we can grow pretty much any protein we want. And in those videos, we talked about how we can take 40% of a total E. coli's protein content and have that be your protein of choice. And what happens is that sometimes these E. coli will put all your proteins into what are called exclusion bodies. That's what's being shown here. They kind of just package that all away. Now, to study your protein, we have to break open these E. coli cells and free your protein. And there are a lot of different techniques to do that. These what are called lysis techniques. Now, at the very beginning of the class, we talked about, remember, hydrolysis, which is water cutting. So there are many different lysis or cutting techniques to burst open cells. And when you burst open a cell, before you purify anything, what you're left with is called the crude extract. Now, to burst open a cell, you can freeze the cell, which will break apart its cell wall. You can use mechanical force to break it apart, or you can use chemical force, like chemicals to break apart the cell wall. If you're working with a tissue, you can use a blender. You can use pressure. Basically, any way you can think of to open up a cell is probably a viable method. In general, you want to pick the most the least stressful method that you can. That is the thing that's going to protect your proteins the best. Now, when your protein's inside of a cell, it's inside its natural environment, so it's good. However, when you bust a protein out of a cell, you need to be worried about many different things. And that is because a protein adopts a natural, most proteins adopt a natural 3D conformation. And they are only active in that conformation. Outside of a cell, a protein can unfold or denature, and then it will no longer work, and your experiments usually won't be worth anything. So some things we have to be aware of. First, pH, and we use buffers. So we talked about buffers already. So your protein will only stay folded at a certain pH. So when we break open a cell, we have to make sure that the crude extract is going into a solution that is buffered to keep our pH balance. We also want to do this lysis at temperatures near 0. The warmer the temperature is, the more likely your protein will unfold. So at 0, we are keeping the temperature low enough that it will make it unlikely to unfold. Also, when we burst open a cell, we are releasing other things than our target protein. We are releasing things that are called proteases. These are enzymes that eat proteins, and nucleases, enzymes that eat DNA. So we need to inactivate these. So we can do that through pH or temperature, or what is often done is that you add another compound to block the action of these compounds. So EDTA is usually the go-to compound. EDTA cleates or absorbs metal ions. These enzymes usually need magnesium or another metal ion to work. We use EDTA to stop these from working. Proteins will also denature if they come in contact with air or glass or plastic. So when we are working with proteins, we have to be very careful not to create bubbles. So if we're mixing things, we have to be very gentle. If we're pipetting things, we have to be very gentle. And when we purify proteins, we may not want to use that protein that day, or we may not want to use that protein this month. And so when we store proteins, we usually do long-term storage at, this should be minus 80 degrees centigrade, so I apologize for that, minus 80 degrees centigrade, and we can store them under inert conditions, because oxygen might denature it and temperature will also denature it. Now, when we're purifying the proteins, we need to be able to detect that our protein is there. And usually, we want to know how much our protein is there. So we use what are called assays to do that. Now, if our protein of interest is an enzyme, this is actually easy to do. What we do is that we allow the enzyme, which you remember is a protein that catalyzes a reaction, we allow that protein to do its reaction. And there are some enzymes out there that will create a product that is either colored or has fluorescence. And so if we can measure this fluorescence or color somehow, which we can determine the amount of enzyme we have. Or another way to do this is through what's called a coupled enzymatic reaction. So if our enzyme of interest produces a compound that isn't fluorescent, well, we can add a second enzyme that works on the product of our enzyme of choice. And that second enzyme can produce a colored product. So our first enzyme, non-colored, our second enzyme will produce a colored or fluorescent product. We measure that, and then we can determine the amount of enzyme we have. Now, if we are not using an enzyme or we do not have a way to make a colored product, we can use what are called immunoassays. That is, we can harness the power of antibodies for our purposes. Two popular techniques are radio immunoassays, RIA, or very popular in biochemistry, are called ELISAs, Enzyme-Linked Immunosorbent Assay. So we're going to go through how an ELISA works. So let's say you are purifying a protein and you want to know your protein is there. So the first thing you do is that you take an antibody and you mobilize it on some solid support. Something that's solid, right? Could be a membrane, and that's generally what it is, a membrane. Now, this antibody is going to bind to your protein of interest. So you're going to develop what's called the first antibody that will be able to bind to your protein. So you're going to wash over your crude extract, and this antibody will be specific to your protein. Then, after that, you're going to use a secondary antibody that is connected to an enzyme. And this secondary antibody can only bind to your antibody protein complex. And there's many ways to do this, and we're not really going to go into it. All I care about right now is that you have the general idea of how this works. So this secondary antibody recognizes your primary antibody and your enzyme complex. And the enzyme that is attached to this secondary antibody will create a colored product. So we add substrate. This substrate will interact with your secondary enzyme here and create a product that we can measure either through radioactivity or fluorescence. And using an ELISA technique, we can not only detect if your protein is there, we can start to measure how much protein, your target protein, is available when you start to purify from a crude extract. And that's it for this video. I will see you in the next video.